I've never been accused of needing a microphone. Hello, everybody. My name is Nancy Moore. I am an instructor at Kwantlen Polytechnic University Brewing and Brewery Operations program, and I was just counting how many students there are in the room. Glad to see you all decided to come to the right talk. What I wanted to start just by talking a little bit about the program. I think most of you know who we are and what we've been doing, but next year is our 10th cohort coming through, and I'm not able to count how many students that means that have come through, but there's at least 10 of them in this room, so that's good. We've been having a lot of fun. I think our students have been having a lot of fun, and I think the most important thing is the impact that I see them having out in the industry. So what I'm going to be doing, anyone that watches me on Saturday night with another little piece of paper, it will be keeping a tab of what percentage of the winners have one of our students involved in it, because that's my measure of whether or not we're being successful. After almost 10 years, we've just finished a program review. And so I'd like to thank all of you that contributed to that helped us understand what we're doing well, what the students can do when they come out, and what we need to change to have them add more value to your business, because that's what this is all about. And I guess to do a brief plug that we have a job board. I'm not sure if everyone's aware of that. If you go to kpu.ca/brew, something like that, something brew, you will find the job board. If you've got a job ready to be filled, you can, any employer can add their jobs there. If you're looking for a job, that's where we hope there'll be there are a lot of brewing jobs up there. So if you're looking for a job, check it out as well. And on March the 6th, we are having our annual career fair. So if you want to meet both the first year and the second year students and see who's coming through, we'll be doing that out at Kwantlen. And I think there are details on the website about it. Not yet. There will be details on the website, so you can sign up as an employer and come out. Whether you have jobs to fill or not, you'll be able to meet the students and find out which are the ones that when you do have a job, you want to go and find and get them to come out. And I'm told there's a video back there someplace I'm supposed to play.
What will it mean to you? For some, it's where the impossible becomes possible and where true transformation takes place. For others, it's the moment when you realize that boundaries aren't meant to be pushed. They're meant to be broken. It's what turns dreams into goals and plans into accomplishments. It's where challenge meets opportunity, and it can be the guiding light in the fog or the spark, igniting the engine that was always there. It's a place to embrace diversity and welcome challenges with open arms and a fighting spirit, where you empower each other and build lasting bonds. A place where experience drives learning. A place to be bolder, braver. Whatever it means to you, one thing is certain. It's where thought meets action. And at KPU, it's only the beginning.
Thank you. The other reason to come out to the brewery is we have the cutest 2 hectoliter brewery you have ever seen. 4 full vessels. It's exactly the same, I think, as Steamworks is running at 50 hex, but ours is much cuter. Which may not be the reason that most people like breweries, but I think it is. Anyway, today we're here to listen to Dr. Matthias Schutz. I've never called him doctor before. And Matthias leads hops R&D in the KPU Applied Genomics Center. Now, my real job today is I'm going to tell you what all these words were that I had to look up on the computer last night and went, what does that mean? So genomics is genetics. Am I right there? Good. Okay. He'll tell me if I'm wrong. Matthias owns as a PhD from Simon Fraser and completed then postdoctoral work in the Department of Botany. and in the Michael Smith laboratories at the University of BC. And here's a little history for those of you that weren't around Vancouver in '93. Michael Smith he died. He was a Nobel laureate in chemistry. And when he won, he donated his money to help other people learn things and cure diseases. And so they named a lab after him at UBC, which I think is pretty cool. So Matthias has more than 15 years of experience in metabolomics. Anyone know what that word is? Me neither. It's the cell cultures, right? Or dealing with the actual cells. All the cool chemistry. Okay. Germplasm development, plant pathogen management. We all know what pathogens are, only when they're in beer. Cultivar development, cultivation of hops and cannabis, high performance liquid chromatography, HPLC, and gas chromatography, mass spectrometry, which is just different ways of separating compounds that are like you have in hop compounds and analyzing them. What else do they want to know? So what their guys are doing is building on modern developments in technology and understanding in the fields of genetics and cellular products. And most of us think about this as being done with medical science, but these guys are actually doing the applied part of it, which is so that they can apply it to practical things like growing hops and making beer. Matthias.
Thank you. Can you guys hear me without the microphone in the back? Is it good? Because yeah, I'm not really the MC kind of person, so we'll put that there. Yeah, thanks for the introduction. And let's talk about hops. That's why we're all here. All those fancy terms for dabble, mix, things like that. There's tools to be used for that. And my talk title is BC Hop Varieties for BC Brewers. So a little bit of introduction very quickly, because I think everybody here is kind of a hop nerd, right? You guys all love hops. You like different kinds of hops. You like how they taste in beer. You dry hops, you add oil. But it all comes down to chemistry. And hops, when you look down, these tiny little cones have. It looks a little bit like a waffle cone. Like a waffle cone that's empty. Early in the season, when the cones are just forming, they're empty. There's no oil inside. And then they slowly fill up like a waffle cone does, like that you get at McDonald's and stuff like that, right? Instead of having vanilla, sugar, or syrup in there, you have a whole bunch of chemistry. And that chemistry is what we harness in brewing in order to make special aromas, flavors to our beer. So there's a lot of things. Bitter acid, you guys are familiar with, of course, terpene, style, fatty acid esters. We have a beer that we can give people to sample at the end of the talk that was made with one of our new varieties that is very pronounced in one of the categories. Very interesting to give them feedback. So what determines hop chemistry? There's basically two determinants. There's the genetic component, makes sense. Different varieties taste and smell different than other varieties. But there's also environmental component, or the terroir, or the farm craft. So we're going to talk about that one first. So first of all, southwestern BC is a great place to grow hops. So if you have abundant water, you have abundant farmland. These are actual pictures from a hop yard that's here in BC. A milder climate and abundant water highlight our growing. So compared to 6 hours south, the Yakima is a very different climate, almost desert-like climate. Or the Okanagan, once you cross those mountains, is much hotter, much drier. You see that pulling some data from Environment Canada and NOAA, the Yakima and Abbotsford, see that in the green bars, we have a lot more precipitation throughout the growing season, and we have much lower temperatures. Right, the average high temperature around 25 degrees in Abbotsford compared to 32 degrees for Yakima. So these are average highs, and of course we know that climate is changing. Climate change is going to impact hop production. First. Oh, for the recording, I have to use the mic. Okay, I have to use the mic after all. Okay. So climate change is going to impact hop production. It's already impacting hop production. if you Google or you go to Google Scholar and you say climate change, hops, hop production, yield, you're going to get a variety of different papers. Here's some headlines. Heat Hops Hallertau, exploring implications of climate change for German beer sector. The impact of climate change on yield and quality of Zaz hops in the Czech Republic. A short water supply in Yakima River Basin could mean less waters for some growers. Not just availability of water, it's also these very, very, very high temperatures. So it is increasingly getting hard to farm anything, also hops, in a changing climate. In an arid climate, such down in Yakima, where the bulk of hop production in the US is centered, there needs to be about 700 to 800 millimeters of supplemental irrigation. So 70 to 800, 700, 800 is about this much. Imagine this entire room filled with that much water, and now put that out to 10 acres, 1,000 acres. That's a lot of water that's being used for hop irrigation. Simulations using future climate prediction all show a decline in hop productivity and a decline also in alpha acid content. So these are facts. This is what the studies are showing us. So I wanted to show you some impressions of some local hop production. We do have a small but very dedicated group of farmers that are growing hops here in British Columbia. They're coming up with a variety of very, very high quality hops, which I'm going to show you some data on. And it all starts from, we have a propagation greenhouse and green flora greenhouses that is making the plants. They're propagating the varieties that can then be grown in fields. The fields are then harvested, not by hand, but using, sophisticated equipment, industry standard equipment, just like you see everywhere else in the world. We also harvest our hops using automatic picking. All hops being grown in British Columbia are pelletized in British Columbia. Every single hop farmer that is growing hops right now has a pellet mill, and they are pelletizing hops in British Columbia on site. And this is a value add because you can get those hops faster. British Columbia also has some unique fertilization. So we have a little bit of visitors in the fall that can come along and bring some of that salmon resources and sprinkle it all over our hop fields. A tremendous marketing opportunity if I ever saw one. So the question I was asked from hop growers and brewers is, are US commercial hops better than BC grown hops? As a scientist, I asked myself, what is better? How do you measure better? I'm not a very good sensory scientist where I can pick out these pineapple flavors or mango flavors. That's just not what I'm good at. What I'm good at is leveraging a team of talented students and professionals to evaluate data, evaluate samples side by side using analytical chemistry. So we use, there seems to be something missing here. Yeah, that's okay. We use standard methods from the American Society of Brewing Chemists or the European Brewing Convention to analyze our results, which means that our results, the way that we perform those results are being done the same way in labs all over the world. So we break down the alpha beta acid content by extracting all of the alpha beta acids out of the hop pellets. We then separate them on a chromatography machine, which we have a dedicated chromatography machine just for hops and brewing science. Just to let you know, like that's all that it's used for. We can get alpha acid breakdown, the cohumulan to adhumulone, total alpha acid, beta acid, and also the essential oils. So using these standardized techniques, we went out to some brewers and we asked, hey, can we have 50 grams or 100 grams of what you got in the freezer? So that's what we did. It's really pixelated. Apologies for that. There's a white paper on this, which I can disseminate afterwards. But what we did is we took what we found in the freezers of brewers, and let's say it was Cascade, and we compared it to Cascade that was grown here locally. When it comes to alpha beta acids, sorry, you can't really see, they're very comparable, right? But when it comes to Fuggle and Srirachiase, varieties that do better in a more temperate climate, we're actually outperforming them. There's more alpha acids that we find in BC hops, as seen by these bars here, compared to these commercial varieties, which I'm not, they were randomized. No one's being called out here on what farm was growing what. Same is true for the BC growers and for the commercial growers. Same is true also for essential oils. So again, for Cascade, British Columbia was actually growing, providing a lot more essential oils coming out of those pellets than we were seeing from the commercial hops. And the same is true for other varieties, Fuggle and Srirachaise. So like I said, all of this data is available. It was sponsored by the industry, but it was done in a way that it was blind and fair in order to not provide any kind of bias. One thing that is a caveat, all of these hops, we did a study in February, March, April, a number of years ago. And I know the hops that we looked at for our BC grown hops were hops that were harvested that past season. Whereas what I got from the freezer, I don't know when that was harvested. But as a brewer, that's still a value add. If you have a supply chain where you can get hops that you know are a year old or that you can get that same year, that's still an advantage for you, especially if you're making lager and all you're interested in is alpha acid content, right? So what about genetics? Everyone talks about genetics, in cannabis, all these different strains of cannabis plants. There's a number of experimental hop plants. Seems like everyone has a breeding program. We are one of those people. We also have a breeding program, but I want to explain it to you a little bit, what makes ours very robust and evidence-based. So genetics, of course, makes a, okay, for some reason, some of my images are not showing up. This could be a Mac to PC issue, so apologies for that. What you should see here is various breeding programs have given us some popular varieties. Cascade from the USDA breeding program. What did I have next? Mosaic, right? Which is from the hop breeding company. And then I had Magnum, which was, of course, from the German breeding program. So these are varieties that have been around for a long time and are very popular in making beer. So the Canadian backstory to Brewers Gold. So Brewers Gold was this very high alpha variety that was actually generated over 100 years ago. And its pedigree is actually has Canadian origins. A wild Manitoba male was used to generate Brewers Gold. And Brewers Gold claim to fame is that it's arguably, it was used to breed notable varieties like Galena, as well as Nugget and Centennial, and is still used for breeding today. So the foundation of a lot of our modern hops have a Canadian origin. So the question that was asked was, can we develop some new hops that are adapted to grow well here and make great beer? This is actually more difficult than you would think, because we have to make farmers happy so that they can make money. So if you have a great hop, but only yields 700 pounds per acre, the farmer's not going to be able to make money, but it might be a great hop for brewing beer. Likewise, if I have a hop that makes 3,000 pounds per acre, but the brewers are like, no, this is not going to work for me, then it's also not a successful hop. So we need to balance both sides of the equation. So we look at both agronomic traits and we're looking at brewing traits. And luckily at Kwantlen, like Nancy had just given us a good introduction to, is you guys are all aware that we have a brewing program. And this is an excellent opportunity for students to be able to be involved in evaluating that process. We've had hops that when we've done the rub and we smell, experimental hops that is, and they smell fantastic. We make beer with them. Dominic sends me a message. There's something wrong with the beer. So, well, was your student? No, it was the hop. It was the hop. It gave a very unpalatable kind of flavor that was not very desirable. So it's not until we start making beer that we really know what the value or the value add of the hop is to the brewer. So why have a Canadian hop breeding program? So Canada imports nearly 2,500 tons of hops for a domestic brewing industry. Those are stats from 2020 from USA Hops. There's a long history of hop cultivation in the BC Fraser Valley. Water availability, milder climate, and abundant farmland all contribute to a business case for sustainable hop production in our region. I talked to the BC Ministry of Agriculture, and they did a quick kind of look, and they identified 16,000 acres just in the Fraser Valley that they could see being converted from a minimal or low yield, low economic yielding purpose, such as forage or just being vacant, that could be converted to value add crops such as hops. In the 1930s, 40s, Agassiz, Chilliwack was the largest hop producing region in the British Commonwealth. So that's like, wow, that's really big. So 600 acres. 600 acres was a lot of hops back in the 1920s and 30s, but nonetheless, it's more than we have in the ground right now. So British Columbia has, via the hard work from some dedicated growers that are been working with me on this program. And to be clear, it's actually their program. So I'm just the kind of middleman. I'm the science guy, the plant science guy that helps develop these varieties, but I have no ownership. KPU has no ownership. These varieties belong to the growers and the industry partners that we that support the research. So the challenge is how to develop highly productive and disease-resilient hop varieties in a changing climate. We have, as brewers, you may not be so familiar, but there are a lot of downy mildew, powdery mildew issues. There's hermaphrodism, where you can have all of a sudden, Cascade is very famous for that, where you have male flowers forming instead of female flowers. Usually those flowers are not going to make pollen, so no worries about seeds, but it does decrease the yield. In 2021, we had this heat dome. Do you guys all remember 2021, the heat dome? Couldn't go outside. It was like just insane. Well, Fuggle, which is an old British hop, it suffered greatly, even here in BC, didn't make it to the top of the wire. Whereas 2 of our experimental crosses did just fine. The pedigree of these crosses were we used a male that was sourced from 3,000 feet elevation near the New Mexico-Arizona border. So we had some heat tolerance traits built into these hops. We need further refinement in order to have those hops make good beer, which is what my job is. But I was really happy to see when there's a natural disaster, a guy like me doesn't say like, oh, that really sucks. It's like, ooh, opportunity to learn, right? So that was really interesting to see our original premise of making these crosses actually hold true in the field. So when people stopped cultivating hops in the 1940s, a lot of the hops just became feral. They started kind of growing along ditches, along the sides of the roads. There's even a little mountain called Little Hop Mountain in Agassiz. So I said, okay, I'm going to go to Little Hop Mountain in Agassiz. And I went there, found a lot of hops, a lot of male and female hops, all a crap. I went up that mountain, it's about 300 meters high, and went down the other side, and there was a ton of diversity of hops there. So we've collected a lot of hops here because there's been an experiment going on the last 80 years about which hops are surviving. So we're having both male and female hops in the field, so we're having sex. So sex is going to generate diversity, and it's going to give us diversity where the more fit hops that are downy mildew resistance, for example, will thrive more than ones that are not. As a side program, or in addition to this program, Langara College has also been working with Barnside Brewing. They just made a tasty beer with using some of these hops directly for making beer. We're using these hops as breeding stock, but of course, some of those varieties, when we genotyped them, we extracted the DNA. Actually, Garrett extracted the DNA because I really don't do anything except sit on a computer. And then Garrett did the DNA analysis, and he said, hey, this is Goldings. And guess what? The Chilliwack Museum shows records that Goldings was grown in that region. And then we extract the DNA of another one of these, maybe the one that we found here in Abbotsford. And it's something new. It's something that's hybridized, something that's had recombined. So we're just using this population to introduce disease-resistant traits. And we're using existing varieties to give us chemistry traits. What we didn't realize when we started out on this process was that actually there's a lot of new chemistry that you can generate just by making crosses. So when we make a cross with Cascade, the progeny often don't smell or taste or perform anything like Cascade. There's tremendous heterozygosity. So diversity is not a problem. We have a ton of diversity. We generate around 1,800 plants, and we've had everything that makes no oils at all. It's completely makes no lupulin glands at all, which is, from a genetic perspective, very, very interesting, because those really bad hops, they teach us, okay, we don't want that. We want the other thing. So even the bad hops are valuable for our breeding program. Again, some things missing on the slides here, but that's okay. When you eat corn, when you eat bread, wheat, barley, soybean, maize, all of these contemporary crops are using genomics in their breeding platforms. That is how they're gaining genetic gain and yield over successive years of breeding. It's much more efficient and much faster. Costs money. So we have and are building a dedicated genomics platform where we can look at the entire genome of hops in a cost-efficient manner. And I'm happy to talk about the details with that, but it's kind of a collaboration with people doing fundamental research and publishing the genomic sequence of hop and continually improving upon that. And now we're taking that information and we're building a high throughput genotyping array, which basically is going to help us identify traits of interest before planting them in the field. More on that in a few slides. So 2018 is kind of when we started doing our first crosses, collecting some varieties out in the field, getting some varieties in, public varieties, Cascade, Chinook, Sriracha Ace, all those varieties that we can get our hands on, and generate some diversity. We screen for downy mildew resistance because, again, the hop that we generate needs to grow really well here. Now we have these growing trials, phenotype collection, and eventually we keep on doing this process until we get to this elite cultivar. The funding of this program ends in 2026. We've identified a mechanism to continue with this breeding program. But what's really been fantastic is that we've had a investment from Genome British Columbia and the Investment Agriculture Foundation, $250,000 to build this genomics platform. This genomics platform is going to help us get there faster. So imagine I have 1,000 seeds in my greenhouse at KPU. And now I have to find an acre to grow those 1,000 plants. I need to hire a student or a team of students to go look at those plants. I need to say, okay, which ones are growing well, which ones have disease resistance. Then we have to harvest them. We have to extract the alpha beta acids, all the terpenes. We might have to brew with them. It's a lot of work. Now, using this genomics platform, I take those 1,000 seedlings. Garrett and his team of students extract DNA from all of them. They already do it already because we check for male and female hops in the lab. And now we'll be able to say, we're only going to plant these 100 in the field because these 100 are going to have high alpha acid content, good downy mildew resistance, and have very interesting oil chemistries. So that is the value of having this platform. So just to give you a snapshot, the students have been busy in the field because we're building this platform now. So we've been doing traditional phenotyping. They're out in the field. They're taking pictures of hops. How do the leaves look? What's the size of the hops? Which one, if you're a farmer, which one would you want to grow? This one or that one, right? So we have, but this one is also important because it tells us, okay, what gives us that sucks at life phenotype, right? Because we don't want our plants to suck at life. So we want robust, healthy, vigorous plants. So we also look at the chemistry, right? We can't brew, even in this beautiful brewery that KPU has, 2-hectare brewery. I can't go to Dominic and Nancy and say, hey, can we brew 200 batches of experimental ales? They're not even going to answer your email. So this is high throughput. We can do the analysis of this and we can get, some good results. So this is Cascade to give you guys an idea as a, like kind of landmark. You can put a pin in it. A little bit higher on the alpha acid content, but it's grown locally, so that's why it's high quality. Beta acid, major terpenes, minor terpenes. Here's the breakdown. Doesn't matter. If you're really interested, I have all these COAs and I'm happy to share. Here's a variety. Oh, something about our naming convention. We don't have HBC 5968. Our naming convention goes 20. That is the year the cross was made. CE, the female was centennial. 01 is the name of the male that we collected or that we have generated in our population. And then -4 is seed number 4. So 20 CE01-4, to be named something sexier later, has good alpha acid content, has a ton of terpenes. Look at that, 2.88% compared to 1.86% in Cascade. Plus, we have identified geraniol. This is, very abundant, relatively abundant, and 2-methyl-butyrate isobutyrate. 2-methyl-butyl isobutyrate. Okay, it smells like mango. Okay, it's very, very fruity. So we have a lot of that in that variety. And the sensory notes were sweet herbal citrus. And that is something that we had gotten before we did the chemical analysis, right? So this kind of validates those initial findings. These sensory notes for this one is pineapple, tropical fruit, very high alpha acid content, again, very high 2-methyl-butyl isobutyrate. We also did some sensory analysis with the KPU brewery, where we're using, which actually, by the way, this is a great segue. After this talk, head down the hall there. You can do some sensory analysis on some local grown hops and some experimental hops. I want to know what you guys think. Maybe you guys will pick. We'll see what you guys pick. It'd be really, really interesting what you guys will pick. I, of course, have my favorites. So we use this again as established methods. We use an ASBC sensory method with 12 panelists where we make a tea of the hops, and then we have a randomized kind of evaluation. Comes down to making graphs like this. This is a variety that was super, super popular, is very, very popular. It's not what we have here. We have a different variety in the beer right now. I want your feedback on that one. This one is super berry citrus herbal. Has good alpha acid content, around 8%. Very, very high yielding, around 2,000 pounds per acre in the second year. So plants mature, right? You get full harvest around year 3, 4, depending who you talk to. 1.5 acres of this hop were planted this season. So reach out to make it easy to the BC Hop Growers Association. And this hop is also in the sensory selection event, but you won't know which it is because it's all randomized and blind. So this hop was we have around 3 or 4 very promising varieties that we're almost ready to, put to bigger field trials like we did with GAN, 1.5 acres. So the whole point of this is to support brewing innovation. Brewing innovation is the heart of all of this, right? We're not growing hops just for the sake of growing hops. Those hops should be used in making beer. The KPU brewery is a perfect partner for this. We have enthusiastic students that want to work with these hops. They want to learn, and they will then bring that curiosity forward after they graduate and get a real job. So here's Ernesto. He was a brewing student that worked with us last year. He's now employed in the brewing industry. There's Dominic down there changing hoses. Alec and Emily at the Vancouver Craft Beer Week last year, pouring an experimental pale ale. So this is the beer that we're happy to give you a taster for. I'd like to hear your feedback. The beer was made 4 weeks ago. It was just bottled by Emily. Thank you, Emily and team. Here's the malt, the grain bill, brewing salts, USO5 was used as the yeast. Here's the hop additions. So as they start pouring, I'll take questions, but please do feel free to take a sample. But before we do, I'll go back to the slide. I wanted to thank everyone. I want to thank Garrett, Katie, and all the students that have been working on this project, and the KPU Brewing team and their students, and of course the support I get from the administration of the Applied Genomics Center and all of the funding. So one last little slide. The Applied Genomics Center wants to work with you. I have dedicated equipment for brewing. I was at a non-alcoholic beer talk yesterday, and they said, oh, I have the advantage of having an HPLC to check my sugars. I have a dedicated HPLC for looking at sugars and wort. For every dollar that industry comes with, I can multiply that by 3 with the federal money in the way of grants. So this is the cheapest way to do R&D. So please do reach out if you have a problem, if you want to optimize a process, if you want to look at isoalpha acid analysis, not by math, by actually looking at samples, come talk to us. Diacetyl, total fermentable carbohydrates, these are just some of the things that we can work on. So at this point, I'm going to go back to this experimental pale ale slide, but I'd be happy to take any questions. And I'm also happy that I kind of rushed through the talk and we're on time, not like the last talk. Ken.
So a little praise and a plea. So my name is Ken Malenstyn. I'm one of the owners of Barnside and also a chair of the BC Hop Growers. And I think I want to just let you all understand that what we're able to do here is at the scale of many of the bigger breeding programs in the world. It's something that we as a small group of growers could never accomplish. So I have to give thanks to Matthias and KPU for everything they're doing for us. I mean, this is a big deal and we're doing it at a world-class scale, and it's hopefully going to be really meaningful for us as a group of growers, but it should be really meaningful to you guys too. So thank you very much for everything that you guys are doing. You're really helping elevate our industry. And I think my plea to you in the room who are brewers is take advantage of this. I mean, I can wax poetic about all the reasons that you should work with local hops, local ingredients. Matthias has hit on some of those, but there's an opportunity here to have some fun. It's craft. It's why we're all in it. And, we would love to get people to, alpha beta test, if you will, with stuff that Matthias and the rest of the growers are doing. So thank you guys actually for being here. I'm stoked to see the room so full. And again, thank you, Matthias and KPU.
More questions for Matthias?
You need one? I don't need a mic, but I'll use it. I had a question mainly about what you're prioritizing for future breeding. Are you looking more at producing new alpha varieties, more climate varieties, or sustainable varieties, or are we looking mainly at creating new aroma varieties?
Sustainability is key. All of our varieties have to be sustainable, which means that they have to be downy mildew resistant and they have to yield a certain pounds per acre, right? One of the very famous varieties is who shall not be named, is very low yielding, right? As soon as the price collapses for that variety, then it's going to be very hard on farmers. So sustainability is a really big key. And growing in our region is going to be much more sustainable in the future. I mean, we all see the ravages of climate change. In Europe, they have issues with hail. They have issues with water. We don't have such issues with hail. We have abundant water. Only 30% of the Hallertau is irrigated, right? So it's a problem. So as far as varieties go, alpha varieties or aroma varieties, we're going more for aroma varieties, and we're going for a more noble character, more spicy, caryophyllene rich kind of hops. And we're also going for very fruity, ester forward kind of beers. So I know if you use a thiolized yeast, you can also get a lot of those fruity esters, but I find they can be very unidimensional and you can get much more complex flavors from using hops. So those are the things that we're working on, and that's what we're prioritizing. But I take my marching orders from what the brewers and the growers tell me. So really, it's up to them. Any other questions?
Yeah, I was just curious about, are you specifically sticking to like the locally kind of landrace wild hops that are, result of previous breeding, or are you looking to bring in potentially like European hops and breed from those as well?
So we do have European hops that are publicly available that we can breed with, like Magnum. Like this is a Magnum cross. So this is a Magnum cross that you're tasting right now. We are diluting out the local contribution, except for disease resistance and maybe 1 or 2 genes for interesting chemistry. So a lot of these males, like this BR male, this is another cross with Centennial. I don't know. It's a male that came from another cross from Centennial. So if you want to do 50% Magnum, 25% Centennial, 25% something from Little Hop Mountain in Agassiz, right? And the diversity we get from one cross is just amazing. If any of you guys have been out at Barnside this summer, you might have seen a whole bunch of pots there, and you'd be like, what a freak show of a hop yard. That all came from one cross, and you have such a diversity phenotype. And that was our training set for our genomics platform, that we don't have to grow thousands of plants in the field, that we can do more predictive genotyping and use fewer resources and get to where we need to go faster.
When it comes to the long-term goals regarding the development of these hops, you're obviously developing them with a fairly public program. And so once these varieties eventually, if and when they do get to market with a marketable name, are these going to stick as public varieties? Are you going to partner to make them more proprietary? How does that look long term? What sort of relationship will we have with American growers and that sort of thing?
So the varieties that we're developing will be optimized to grow here, right? So there's going to be advantage to growing them here agronomically. That is what we're going for. We're trying to develop hops that we can grow sustainably here. We are using a variety of different, varieties as our breeding stock. So other hop breeding, like the hop breeding company, they have as their parentals basically what was publicly available from the USDA, right? So we have access to those resources as well. And without those publicly funded resources, there's no way we could have our breeding program, or no one else could. We went the additional step that we went back to the wild, right? So that is something that I think is unique in our program. It also makes it a much more ungainly beast because we get so many diverse phenotypes, but we can also come up with something really new. And we need to reinvent the wheel here for hop cultivation if we want to do it in a changing climate. So we need some more puzzle pieces, and that's what we've tried to incorporate into our program. The last question was, are these going to be proprietary varieties? I actually have an opinion on that, but it's not my opinion that matters because I don't own these varieties. The industry is providing cash, and their in-kind contribution, which we leverage for federal dollars. And the intellectual property belongs to the industry. I have a handshake agreement, and I have talked with the industry that the whole goal is to have and reignite BC hops. I don't know if anyone would want to grow these hops somewhere else. They might not do economically well, but they might. But my hope is to see, I always say the number, don't get scared, 1,000 acres by 2030. Which if the government gave us a tax break on using local hops, I know we'd be there next year. So we also have to do leveraging the government to help us out with this.
So kind of based on that, I'm Melanie from Locality Brewing. Are you trying, if one of the goals is to maximize the unused or not entirely used farmland. Are you looking at the different soil types and microclimates and trying to see what different varieties could be most best grown in different soils and areas?
Great question. Almost like it was planted, but I know it wasn't. Our trial sites are Myrtle Meadows Farms up in Pemberton. If you've been to Pemberton, it's volcanic ash along the Lillooet River, very different kind of climate. Barnside Brewing, Crescent Island Farms, very much more clay rich. They have a unique terroir. And then you go out to the Fraser Valley, Bradenhoff Hops, Tops Hops. There are partners out there. And when they send us their samples, we do see some differences. To sum it all up, the climate, temperatures, and water are more similar than they are different. Soil does seem to have a big impact, but not as big as just maximum nighttime temperatures, maximum daytime temperatures, and water. Irrigation, crown cutting, training dates, those are all part of farm practice, and they do impact your final product. If Mark Topper and Ray Bradenhoff, their farms are close together, if they do a different crown cutting, if they do different training dates, they do different harvest dates, they're going to have a slightly different product. And down at the MBAA meeting in Seattle 2 weekends ago, they had, was it Amarillo? Amarillo harvested at 3 different times, and it was very, very different. So the big hop companies, of course, they have thousands of acres to harvest. So blending and normalizing is big, but you guys can take advantage of this unique terroir of that these different farmers offer. I really think that is a value add, especially if you're craft, right? For craft brewers that want to capture those differences from those regions.
Is sort of a two-part question. You were saying that the soil doesn't really have as much an effect as the environment itself. Is it more of a pH change between the soils, and then the second piece is, I know that there's some natural landrace hops from the East Coast that are being cultivated in Prince Edward Island, and I was wondering if there's any kind of collaboration that you guys are looking at with that.
I wish there was more collaboration. I've reached out to everyone that I can in Canada. I've talked to Yakima Chief. I've talked to John I. Haas, Hopsteiner. They're all much bigger than us. They have their own breeding program, and they're going in that direction. They're well-wishers for sure. I haven't heard about the East Coast program. I have talked to, maybe Ken can say a little bit about that. I have recently reached out to all the main hop grower, like the Ken Malenstyn counterparts for the BC Hop Growers Association for Ontario, Saskatchewan, to try to say, hey, what are you guys doing? Let's try to get a conversation. But I tried that 2 years ago and I didn't get much of a response. So I'm hoping I did get a response this year. So stay tuned. It'd be nice to be more collaborative. I'm all about collaborating. So Ken, did you want to comment?
Yeah, I think just we are, I know on the feral hop program with the wild hops that we are working with Josh Mayich, and he's in PI. But our goal with that is to grow a Canadian hop growing industry. So we are having informal conversations with growers in Saskatchewan, Quebec and Ontario, Maritimes. That'll be important for us in the long run to build that, national body that will be, made up of members of all the provincial organizations that are out there, whether formal or informal. So we're working on it. It's just, everybody's farmers too, so we don't have a lot of time, but we're doing the best we can.
Okay. I'm happy to chat with anyone over a beer, especially, or at the BC Hop Growers Association selection event just down the hall. Really, really important, I think, to check that out. All of the hops are loose leaf, but they are pelletized here. You will receive beautiful pellets directly from the farms if you place an order. And I think you will be pleasantly surprised to look at BC hops again, because I know some of you that have been in the industry for a while may have had some difficulties or challenges with hops in British Columbia 6-7 years ago, but these dedicated group of growers that I'm growing with and what they're growing, it's really good stuff. And I think they're no nonsense kind of guys. They will deliver.
That's all the questions. It's almost time for lunch. Matthias, thank you so much for coming today and demystifying what you're doing and helping us all see the results. That was a great berry flavor on that beer. That was quite amazing. I may steal one of those and take. Can I steal one to take home? Anyway, and just thanks to everybody in the hop industry that's working on helping us rebuild the BC hop industry, because I think it is a strategic advantage that we can point to and build on to help build the brand of BC beer. So thank you so much. Now that we should have lots of these for all your people, but you can carry your beer home in that one. Thanks for coming, everyone.